Hi, I'm Angie Patterson. I'm the Master Science Educator here at BlackRock, and I'm also a plant ecophysiologist. So I study how plants respond to climate change physiologically. And I do that by measuring photosynthesis and respiration, which are two processes that help plants grow. So today we are going to explore a little bit about why we're measuring photosynthesis in trees to better understand how these forests are growing, to measure their growth on different levels, and to understand how plants will be um, responding to a changing climate in a warmer world. So stay tuned. Right here, we're at the fire tower, and we'll be exploring the area around the fire tower to learn more about these things.
Good morning, everyone. My name is Grayson Badgley, and I'm a researcher here at BlackRock. And today we're going to talk about photosynthesis, and we're going to talk about how we study photosynthesis at the sort of ecosystem scale. And we're going to walk through three different things. We're going to talk about some instruments that we have on this fire tower that you can see behind me. There's actually a camera up there. We're going to go up there a little bit later, and we're going to look at that camera. And that camera is overlooking a study site that we're going to also visit. And what we're going to talk about is how we use measurements at the scale of towers, at the scale of individual trees, and then at the scale of individual leaves to sort of scale our understanding, to take our understanding of photosynthesis that happens at that chloroplast, right? Where water, light, and carbon dioxide are all combined at photosynthesis from that site of photosynthesis. How do we take those patterns and sort of study them at the whole ecosystem scale? So that's what we're going to be doing for the next little bit. All right, so the question that I spend a lot of time thinking about is how do we take measurements of photosynthesis at the scale of a leaf and take that understanding and try and figure out how much photosynthesis is happening across the entire forest. So let's just kind of remind ourselves of what are we talking about when we're talking about photosynthesis. There's a little thing inside of this leaf called a chloroplast. And that chloroplast, if you give it a little bit of light, if you give it water, and if you give it carbon dioxide, it's capable of what we say assimilating carbon, right? There's that molecule rubisco you might remember that you've learned about, that protein, that it takes carbon dioxide, it splits it apart, and then that carbon that it gets from carbon dioxide is what is used by the plant to grow. And so that all of that chemistry and all of that biology that you've been studying is happening inside of this leaf, right? It's sort of this molecular level thing. But then sort of we have to sort of take that cellular level understanding and get to an individual leaf. And then after we have it at one leaf, we now need to get to the whole forest. So what we're doing here is one way to try and get at that kind of going from the leaf to the forest jump. And what we do is every single week I come out and I collect the total number of leaves that fall off of the tree. And I take those leaves back into the lab and I dry them out in an oven. And then after they're completely dry, so there's no water left, I weigh them. And using just a little bit of math, I'm able to translate from the total weight of leaves that I have to the total number of leaves that must be in the forest, right? So basically, if you've got more of a higher weight, that means there's probably more leaves, and we can be a little bit more precise with that using some equations that people have developed. And what that lets us do is it lets us figure out the total number of leaves that are in this portion of the forest. So I've got this bin that's here, but if you actually look out, if we're sort of looking in the understory, we've got about 7 or 8 other bins that are sort of in this one part of the forest. And that means that if I know something about the photosynthesis of any one leaf within this forest, I can then take my measurements of the total number of leaves and rather than being constrained to talking about photosynthesis at the cellular level, or being able, having to talk about it strictly in sort of molecular or biochemical terms, I can start to talk about it in a more holistic or ecosystem scale sense. Alrighty, so I wanted to share one other measurement that we're making sort of at the scale of individual trees before we get up to the tower, where we're going to sort of look at the canopy and the forest as a whole. And what we're looking at here are actually these light sensors that I've installed. And this light sensor is at the bottom of the canopy, and if we actually look up, I've installed the same sensor at various levels throughout the canopy. And what these sensors do is that every single minute, they measure how much light is being received sort of at the top of the sensor. And what this lets us do is we can basically say is that if less light is hitting the sensor, there must be more leaves above it, right? Because the leaves are up there capturing the sunlight. So if the leaf captures the sunlight, it's not going to hit the sensor. And so by having these sensors at various levels throughout the canopy, we can have another way of measuring how much leaf area is there. So this is an approach that complements what we're doing with our leaf collection and our leaf buckets. It's actually a really interesting little setup here. So these little sensors are waterproof and windproof, and they've been out here all year long, and just they're making one measurement every single minute. And we can sort of look up in the tree, and they're all connected into this data logger. And all a data logger is a little computer. It's a little computer that is connected. It's like your cell phone. It's connected to the cell network. And it's got a little solar panel on top of it, and all that thing does is it's just sort of every minute it tells each one of my sensors that I've got to make a measurement, and then it takes that data, collects it, and just sends it to me almost like a text message. And so it shows up on my computer, and then I can use that data to sort of monitor what's happening in almost near real time, once a minute, what's happening in the forest. All right, so we're now almost the top of the Black Rock Forest Fire Tower. And apart from being an absolutely beautiful view up here, hopefully you can sort of see the beautiful rolling Hudson Highlands behind me. We can start to talk about the last scale of sort of what happens at the whole forest level with these measurements of photosynthesis. And just down here, I'm pointing to essentially a video camera. We call it a Phenocam, which is short for phenology camera. And what this camera can do is it can actually tell us how many leaves there are across everything that it can see. So it's capable of measuring variations in light to allow us to understand, again, how many leaves there are at an even larger scale beyond what those buckets can tell us. And so here we kind of have layered several orders of measurements on top of each other. Right? We started with chloroplasts. We sort of started with this molecular level, or this cellular level would probably be a better way to put it. We then talked about the leaf level. We've got the buckets, we collect all the leaves. That lets us say something about sort of that bit of the forest where those buckets are. And now we've got a camera that is looking at an even larger area. And those buckets are down there, we just can't see them right now. And so when we layer all of these measurements on top of each other, we all of a sudden are able to say things about the productivity of this forest at even larger scales. And I think it's sort of here when we're looking across this landscape that we can really start to appreciate why we want to know things about photosynthesis, and why it's really interesting to study photosynthesis at the cellular level, which is what I think a lot of you have been focused on recently. Is that why do we have ecosystems like this? Are they going to keep looking like this in the future? And the only way that we can answer questions about about the health of a forest like this, if we really understand what causes them to grow. Are they growing? Are they healthy right now? What might threaten them in the future? And so we really need to have a handle on this sort of the amount of carbon going into the forest, the amount of growth going into the forest. We need to have a handle on photosynthesis if we want to be able to answer questions at this larger scale. That's sort of the conclusion of our video today. And just to recap, we've talked about photosynthesis across a whole different levels of scale. We've started reminding ourselves about what happens at the cell level, what happens in the chloroplasts, what happens with things with proteins like ribisco, so this sort of bread and butter biochemistry of where photosynthesis actually happens. Then we looked at how how those photosynthetic processes are embedded in individual leaves. We talked about measurements. How do we measure how many leaves are in one part of the forest? We talked about measurements that we're making here to measure leaves using these cameras, so that we can see the total number of leaves at an even larger scale. And in a companion video, I'm going to walk you through some of the data that we're collecting with these Phenocams, with these cameras. And in fact, it's not just here at BlackRock that we're making measurements like this. There are hundreds of locations around the world that are making similar measurements. And what that lets us do is it lets us start to have a grasp of photosynthesis at these larger and larger scales. So we can study things like how healthy are forests in this part of the world versus that part of the world. It's by making these measurements at multiple scales that we can start to understand these processes that drive global exchange of carbon and water by plants.